Welcome everyone to another episode of Whose Gene Is It Anyway? I am your host, as always, Justin. I am excited for today's show. I think it's going to be a good one. Aren't they always? But anyway, today is going to be a really fun topic. We're going to be talking about de-extinction, cloning, lab-grown meat, bioremediation, fixing ecosystems, all kinds of stuff. So I think it's going to be a lot of fun, and we're going to get into it in just a moment. As you can see, there is a donation bar down below here. That is to continue to raise money for our ongoing project of growing human neurons connected to a computer. And today I have a very special treat to start us off. This is the very first new prototype neuron electrode array. So I don't think the camera will focus very well. It's kind of hard to see. But if you want to see a better picture of this, there are some brand new pictures up on Instagram. So links down below if you want to see those. I'm pretty excited. So yeah, if you want me to answer a question while we're going through the presentation today, feel free to donate by using the link below, the Streamlabs link. And yeah, then I'll answer your question while we're talking through some stuff. So anyway, neuron array, very excited. Neurons coming soon. All right, let's get into it. All right, today we're talking about de-extinction. So this is what we're going to talk about today. De-extinction, what is it? Why should we do it? How does it work? Animal cloning, does it work the way it does in movies? Exploring the basics of the process. And then lab-grown meat. Is it a good idea? How it works? All that kind of stuff. So where did this idea come from? Well, if you have been following the news lately, you may have seen an article about mammoth meatballs, which it's a very catchy headline. They have these very dramatic pictures, but what is the reality versus what they actually did? So there's, like I said, there's all these like really super dramatic things, and they're like, well, it probably doesn't taste like woolly mammoth. Yeah, the reason it doesn't taste like woolly mammoth is because that's not actually what they grew. So what they did was they took sheep cells and they took a protein, or the code for a protein from woolly mammoth, and they put that protein and expressed that protein in sheep cells. Now, this isn't particularly, well, interesting, for one. It's very click-worthy, but at the end of the day, they've basically just made sheep cells with one extra protein, which is exceedingly easy to do. If you've seen any of the other episodes that we've done, you'll know that modifying things to express one new protein is super, super easy. And you're actually going to see us do that in an upcoming video very shortly, where we're going to be taking mouse cells and getting them to express proteins from a variety of different places, tardigrades, and coral, all kinds of stuff. So yeah, it's an interesting concept, and it's very click-worthy, but it's not really de-extinction. It's not really, lab-grown mammoth. But so what is de-extinction? Like, what is it really? Basically, the idea is you take something that is extinct. As long as you have a specimen of the extinct animal, it is theoretically possible to bring it back to life. It's not easy, mind you, but it is possible. The examples that tend to be given are things like the woolly mammoth or the dodo or the Tasmanian tiger. Like, these are the really click-worthy ones, but they're also the least interesting to me and honestly to most ecologists. The problem with things like the woolly mammoth or the dodo is they've been extinct for a really long time. Like, even if it was only 100 years, something that's been extinct like many, many decades, the ecosystem moves on pretty quickly, and also changes. Like, the reason these things went extinct is for a variety of reasons, but a lot of it is habitat change or habitat loss. So you can't just grow new ones and put them back where they used to be and hope that it's going to just work out. One of the other ones here is the blue-finned walleye, which is a species that was previously extremely common in the Great Lakes in Canada and the U.S., but it was basically just driven to extinction through invasive species and damage to the ecosystem. So, yes, you could theoretically make one, but where are you going to put it? right? Like you can't just put it back into the ecosystem it came from, because that ecosystem is destroyed, basically. So you would have to first reset the ecosystem, then put the fish back. But that's rarely how it's viewed. So what do you actually need to de-extinct something? Well, I mean, really, it's two things. The first is high quality specimens. You need to be able to get as clean of a DNA read as physically possible in order to be able to get like all the code that you need. So that way you can then recreate that code and put it back into a living system. And then the second thing that you need is a compatible host or some other method of growing the new organism, and we're going to talk about that in a little bit. So just we got a quick donation from SpaceTot 101. So thank you, SpaceTot. It says, how does genetic modification work? Would something super custom be possible, like a fluffy snake, a watermelon that's blue, etc.? I would recommend you watch our previous streams, because then you'll see how easy genetic engineering is, and some of the things that are possible. A fluffy snake would be very hard, though. Anyway, back to de-extinction. So there are a couple of companies that are working on this. 𝐎𝑚 is the one that did the mammoth meatball. But the company that's actually trying to actively de-extinct things is called 𝐎𝐫𝐢𝐠𝐢𝐳 Bioscience. And so I need to be careful in how I describe them, because I think it's a lot of wanky nonsense that is being used to like extract large quantities of money out of investors. But that's not to say that the science isn't good or the scientists they have aren't good. But as a rule, if something is advertised as a George Church company and you're very excited about a genetics thing because George Church was involved, that doesn't actually mean anything. Like they slap his name onto all kinds of stuff these days to basically raise $1 billion to do nothing, over and over and over and over again. So I would take it with a grain of salt. I mean, it's potentially very cool, but it's also just the most vanity project that you've ever seen, for exactly the reasons that I just talked about, which is these organisms are useless. Like, they do not serve a purpose in the ecosystem presently, so it is totally just a rich people vanity project. But that's not to say that the technology isn't useful, right? If you pick more reasonable targets, it's a very useful technology. Whereas if you're just focusing on mammoth, you're basically making toys for rich people or food for rich people. But we'll get to that. So yeah, why de-extinction? And it's the things that I've already been saying, which is zoo curiosities. So, yeah, you could have a Jurassic Park type dealio. Maybe not Jurassic Park, because dino DNA is usually so degraded that you can maybe pull out a gene or two if there's like protein remnants. So you can like sequence the protein remnants and reverse engineer what the DNA might have been. But there's not like a complete genome. So it's more like you could have woolly mammoths, Tasmanian tigers, dodos, this sort of thing. But it would only ever really be zoo curiosities. I mean, ecological restoration groups would fight you tooth and nail if you attempted to put one of these things just back out into the world. But also they don't serve the purpose that the company claims that they do. Like, you can't fix an ecosystem by putting mammoths on it. That said, mammoths are very cool. It'd be nice to have some mammoths around, but it would basically be stuck as a zoo curiosity. The other option is it's exotic food. It's with the rise of lab grown meat and these sorts of de-extinction technologies, yeah, rich people could pay to eat mammoth. And it's like a gloating thing or whatever. Which isn't really that like that's not a reason to get that excited about it, but it's not. I mean, it's not interesting, but it's also not something that you'd find in like Whole Foods. Next to the kangaroo and the ostrich, there's just some mammoth, or maybe there would be, but like beyond that, it's kind of the limit for these super ancient organisms. Whereas if you're talking about things that were very recently extinct, like say in the last 5-10 years, the habitats probably still exist. And the ecosystems probably could still benefit from reintroduction of those organisms, even if it was in, like, say, the past 100 years. So, for example, there's and we'll talk more about this at sort of near the end, where we talk more about ecosystem restoration. But there's examples of reintroducing wolves to areas and having the ecosystem rebalance because they were missing their top predator. But that makes sense if the thing is recently extinct. The other way that this could make sense is just having like ecosystem backups. So if you think that an ecosystem is under threat, you could go through, sequence everything, and ideally take tissue samples of everything you can get your hands on and bank it so that if that ecosystem was ever destroyed, theoretically you could rebuild it from scratch, and put all the correct stuff back in the right places and have it sort of reset. But that's, I mean, the engineering would be on a scale that is unseen to this point in the technology. And then the other is if you have the ability to arbitrarily make animals basically from goo, that actually might make more sense for shipping animals out into space. So be it for livestock or whatever other reasons, like if you're terraforming Mars or something. But we'll get to that near the end of the presentation. Okay, so let's talk about how you would actually do this. Right? But if you want to de-extinct something, you have to, the first thing that you have to be able to do is clone, because you do not have the animal to start with. So you have to start with some other animal and do a little switchy switch with the DNA and have its DNA put into a host's cell, take over, and then be able to be grown into a full animal. So this is the basic way that the cloning works. You start with a sample from the animal that you want to clone. In this case, you'd have to make that cell basically from scratch, and we'll talk about that in a bit. But let's say that you have saved tissue samples from something that's about to go extinct, rather than something that's fully extinct. You save the tissue samples, and you have fibroblasts or just any kind of tissue works, really. Then you take the oocyte, so an egg, from a donor or like a donor organism. So if you're doing something that's extinct, then you want the closest relative, ideally. So if you're doing mammoths, you would want elephant eggs. So you get the elephant eggs, you remove the nucleus from the elephant egg, and then replace it with the nucleus from the tissue sample that you've grown, and then fuse them together and basically kickstart the process, and it will actually, and then you have a now growing embryo, which you can then implant back into a host animal. So in this case it'd be elephants, and then you grow a mammoth. Theoretically, it's basically as easy as that. Obviously, this whole process is quite complicated and difficult to get right, but this is the basics of the process and how it would work. So what does this actually look like? Because people talk about cloning all the time, but it's very rare that they show pictures. So this is the basic apparatus that you need for cloning something. It's an invert microscope with 2 micromanipulators and microinjectors. These are things that you could theoretically DIY. I wouldn't necessarily recommend it, but it is a thing that you could do. But that's basically it. It's 2 pokey things and a microscope. So when you actually look at it stepwise, this is the process of cloning. So you start with your donor oocyte, which still has all of its DNA and such in it, and you first rotate it until this little blob. Hopefully you can see my mouse there, but there's this tiny little blob just a little bit below and to the left of the microneedle, and this is called the M2 spindle. So this is basically where all of the DNA is hanging out right now, or rather, it's actually right here. Sorry, this is the polar body. This is the thing right here. So then these little cells in B, C, and D are just fibroblasts or whatever other donor cells that you want. So this is where the nucleus so this is where your woolly mammoth cells would be, right? So basically what you do is you suck up one of the little cells into the needle and then spurt it back out and then suck it up and then spurt it back out a couple of times, which basically shreds the cell but leaves the nucleus basically intact. So then you suck up the little nucleus and then you go back over to your egg and you push the needle all the way into the back and then inject, and in sort of like a smooth motion, you inject the new nucleus while retracting the needle until you get to, do you see this little blob here? That's actually the M2 spindle. I was mistaken before. So you bring the needle back out until you get to the M2 spindle, and before you pull the needle out, you suck the M2 spindle up and then suck it out. And basically then the last step is you've got to close up the little hole. And the way that you do this is with just a pulse of electricity. then it just reseals, and you now have a complete egg. So basically then what you do is you repeat this several dozen times, so you have as many eggs to work with as physically possible, but then you basically have an embryo ready to go. So all that has to be done is to implant this into a host, and theoretically it would grow into a mammoth. Now, the reason that cloning is hard is not actually because the process itself is difficult. Like, this bit is actually fairly straightforward. The reason cloning is hard is because it rarely works properly. So for the number of eggs that you implant, the number of actually like healthy, viable offspring that you get is very low. So you end up with like a lot of like malformed stuff and, you know, things that just don't work properly. So you end up having to just do this over and over and over and over and over again until you get that first viable organism. So like Dolly the sheep is one of the really, is one of the really famous examples of this. It took a lot of sheep before they got Dolly the sheep. And this is sort of the reason why we don't do human cloning. I mean, A, it's just frickin' expensive, but also you end up with a lot of dead babies. It's pretty brutal. So it's not a process that people are really comfortable with. And then there's the legal and ethical and all the other stuff. So it's a bit iffy. So this is just another little view of sort of what this looks like. It's basically what I said. You know, suck out the spindle, put it in a new nucleus, give it a zap, and let it go. Another example is you don't necessarily need to use an oocyte from the same organism. So in this case, this is showing taking the spindle out of a mouse oocyte and putting it into a pig oocyte and vice versa. And that can make some very weird, you know, chimeric stuff. I don't think anybody's attempted to grow, like, I don't think this is viable. I don't think it grows into a full thing, but this is much closer to what it would be like for de-extinction, because you don't have, you know, mammoth eggs. But what happens if you don't have oocytes? Well, you can actually grow them. You know, this is one of those things where something that people are often told is that, you know, when women are born, they have the number of eggs that they're going to have. When, recent evidence, this is not true. You keep growing more. And if you stimulate the right cells, then you can induce the formation of oocytes from stem cells. It's tricky, but it is possible. And so you could just so if you had your mammoth cells, you could induce them to turn into stem cells and then induce them to turn into oocytes and then go through the whole process. So you could build it all the way up from scratch. So yeah, now you've got your embryo. The next thing is you basically just have to implant it into a healthy host. So in this case, it's showing mice. You know, I wouldn't recommend trying to grow a mammoth in a mouse. It's not going to work very well. But you know, that's the idea. And you mostly just kind of stick it in there and let it go. And then whichever ones attach properly are the ones that end up growing. This is another option if you don't have a womb available. Potentially, we might be able to do it artificially soon. So this rotary culture was able to grow mice up to 11 days before they stopped it. And really, the only thing that stops them from growing it up to a full mouse is they need to be able to connect a blood supply at the point where they grew it. So they're getting very, very, very close to artificial wombs, which I think is just fascinating. And not only were they able to grow it from, like, they were able to take an embryo out of a mouse and then grow it in this system, they were also able to start from stem cells, like pure stem cells. They didn't make eggs and sperm. They just went straight from stem cells directly into embryo, which was really quite impressive and pretty wild. And then they were able to grow things that look exactly like normal embryos, which is pretty crazy. So the technology for artificial wombs and cloning is actually getting much better, which is sort of what's making efforts to de-extinct things sort of viable. This is another option. This was from a few years ago, where they started a little bit later in the process. So they waited until the embryo had developed like the what's it called? Come on, brain fart. The like the feeding tube. Doesn't matter. Anyway, the umbilical cord. There we go. Sorry, a bit slow today. But they waited until the umbilical cord had formed in the embryo before they removed it from the original parent organism, and then they were able to transfer it into this bag system, and were able to grow it to completion from there. So we have two different technologies. One that is able to grow the initial embryos up to the point of needing an umbilical cord, and we have technology that goes post umbilical cord to complete organism. So now you just kind of have to put the two together, grow the little carousel of embryo up to the point where it has needs an umbilical cord. You connect an umbilical cord to it, then you move it to the bag system, and you now have essentially you've grown an animal completely without a parent, which is pretty wild. And if you do this entirely from stem cells, you've essentially just made a whole organism from a single cell, which is pretty wild. Okay, so just a really quick aside about TV and film, just because I found this really interesting. So this is for a scene or some screenshots from a scene from the TV show Invincible, which I personally love. This is not sponsored. This is just very cool. And one of the characters ends up cloning themselves, and what they show is remarkably like what the real process was. And this was actually what prompted me to look up how cloning works the first time. Because, it's the thing that we'd heard about it many times. I'd heard about it while I was in university briefly. But as sort of a curiosity, as like, this is a thing that they did. But I never really knew the process. But this show, after seeing this scene, it was kind of like, does it work like that? And yeah, it works pretty much exactly like this, which I found very impressive. So that's just a fun aside, really. Okay, now so for cloning for de-extinction, there's kind of a couple other problems. One is you don't have the initial tissue necessarily. Like if you're going for a mammoth, you definitely don't. Whereas if you're going for something that isn't quite extinct yet or is very recently extinct, you maybe have a sample already. But if you don't, you have to build the entire chromosomes from scratch. We got a donation. Oh, Space Top, thank you again. If someone's interested in genetic modification, wants to do stuff like that, work with genetic modification, etc., where would they start? How would they get into genetic modification work area? I would go to university for biochem, molecular biology, synthetic biology, that sort of thing. It's your best way to get into it. And as for finding jobs, I mean, that's that the joke with most biograds is, what are the main jobs for a biograd? Making coffee. Unfortunately, there's a lot more biograds than there are bio jobs, but that is rapidly changing. Biology today is where computers were about 30 years ago, so it's going to be the next big frontier. So it's better to get in now. But anyway, back to printing chromosomes. So if you're going to print a chromosome, it's a huge undertaking, even just to print one. And so this is an example from a paper where they printed an entire yeast chromosome from scratch, because yeast only have the one, usually. Well, it depends on the species, but a lot of them only have one big chromosome. So if you print the whole thing, you could just do a little switchy switch and replace the one that's in the yeast, and then you could have a totally de novo synthesized chromosome. But for something like a mammoth, there's a lot more chromosomes, and there's a lot more to do. So you kind of have to do this over and over and over again. And so basically the way that this works is you print little fragments, which you then stick together into bigger fragments, which you stick together into yet bigger fragments until you've built up the whole chromosome, which is a huge, huge undertaking and extremely expensive. It's a lot of code to print. And I mean, even if it's like 9 cents a base pair, if you've got to print 3 billion base pairs, it's a lot of money. So it's quite the undertaking. But once you have your printed chromosomes, then you can start inserting them. So the way that you do this is you essentially, there's a technique called microcell suspension. So you essentially break a cell into individual chromosomes, or you're making these micro pockets of mostly a cell with exactly one chromosome in each little fragment. And so this way you could, have your one chromosome from your first mammoth chromosome, and then you fuse it to your recipient cell, and then through division you're going to end up with some of the daughter cells have the correct chromosome and some of them don't. So then you isolate the ones that do, and then you start the whole process over again. You isolate another single chromosome, fuse it in, and then isolate the clones that have exactly the correct number of chromosomes and type. It would be an enormous undertaking to replace every single chromosome in something, but it is doable. Like, I think that the thing I want to reiterate the most throughout this whole stream is that while all of these technologies would be very, very expensive and extremely difficult, none of this is impossible. It's hard, but it's not impossible. It would just be a lot of work, and it would probably take you, 10 or 15 years to really do it properly. But it is doable. It is doable. So now, what do you do with your like, once you build your first cell, right? Like, once you have your first collection of cells of that new de-extincted organism, what do you do with it? And the answer is you kind of have 2 or 3 options. The first is find a host. Like, if you're going for, I'm going to make a full animal, you either find a host or do the artificial womb thing or whatever, and you grow a full animal. Or option 2, you go lab-grown meat. Now, lab-grown meat is something that's becoming more and more common. I don't think it's really available in stores yet, but it is getting to the point where it's going to be very soon. And there's a lot of technical challenges with it that they've mostly solved, but there's still a lot that they haven't. So it's probably not as great as people bill it, but it's also not as bad as people bill it. It's definitely interesting. But one of the markets for this, it's great to grow cow, but one of the ways to help drive interest and attraction is exactly what Vox did, which is you start producing proteins, like the mammoth protein sheep thing. If you could grow full mammoth cells and make mammoth steaks, the weirdness of it could be a selling factor, and you could charge way more. So you could build the funds to have more cheap options like, cow available. But it's always going to be kind of a niche product. But for growing lab-grown meat, it's not a particularly difficult process. If you not our latest video, but the video we did the month before that, so in February, I think, we actually showed how to grow animal cells in our video. And basically, the difference between this and lab-grown meat is just scale. So we grew very small amounts because I was just doing, sort of regular tissue culture stuff. But the process is essentially the same. You just do more of it. So you grow way more cells, way more liquid. You'd probably do it in suspension culture if you can. And then once you have that, you seed a scaffold with the cells that you want. And so this is sort of what this looks like. You have all of the different cell types. So you can start with a biopsy, isolate stem cells, differentiate them into all the different cell types that you want, or the progenitors of the cell types that you want, and then grow them, seed them onto a scaffold, and then let it grow. And then you've got something that's basically the same as meat. Ayrton just donated. Thank you, Ayrton. Is the technique used by Cartman from South Park to clone a pizzeria more advanced? I do not know. I don't watch a lot of South Park, so I'm not really sure. Sorry. But yeah, so that's basically what lab-grown meat is. So it's not that difficult. At some point on the channel, we'll probably do something like this, or not probably. We plan on doing something like this. Right now, we're looking at turning bread into meat, and also Rice Krispies into bones, because it sounds fun, mostly, and I want to know if they snap, crackle, and pop. Yeah, so it's a pretty straightforward process. So really the only difference between what they're already doing and growing a mammoth is just that first step where you have to do all of this stuff, where you print the chromosome and do the thing. I saw some questions in the feed that are basically asking how chromosomes are printed, and the way that that works is, like I said before, you print little short pieces of DNA. So there's DNA printers. You can just buy them, or you can just pay to have it done, and they can print up to about 180 letters long. So you basically have to print out tens of thousands of little pieces that are 180 letters long, and then stick them all together carefully in the correct order so that it all becomes one big chromosome. So what you would do is you'd print out, 100 sections or whatever, stick those together into bigger sections, and then put the bigger sections together until you've got a whole chromosome, which is what they did here. It's a lot of work, but it's possible. But yeah. Oh, there we go. So yeah, coming to a supermarket near you could be mammoth steaks, because that just might be the only way to get the technology out there. But it is possible. There is the potential that this might be the future we get. This is from Transmetropolitan, one of my all-time favorite graphic novels. And in their stores, you can buy anything you want: human meat, caribou eyeballs, squid tentacles that are grown extra big, whatever you want. They have just their technology is essentially peaked, so they can do stuff like this with very little effort. So yeah, one day you might be able to go buy a bucket of caribou eyeballs at your local Whole Foods. But that's pretty dystopian. Like it's pretty drab and dark to think about, like the only uses for de-extinction being, just food. It's very grimdark future, which is not my favorite. So I've been playing a lot of this game lately. It's called Terra Nil. This is again not a sponsor. I just really like the game. And it's basically just a city builder where instead of building cities, you build ecosystems and then remove all of your presence from the land. I really like it. But it's basically been making me think a lot about ecosystems and rebuilding ecosystems. So in areas where there's been a lot of clear cutting, or the coast has been destroyed, or whatever the case may be, rebuilding ecosystems, I personally think, is kind of the best use for this technology. And it's also like the most viable. Like, if there was ever a point of de-extincting something, it would be to de-extinct something that was just extinct. Like, we couldn't get our shit together well enough to prevent the event happening, but at least we can like mop up the mess afterwards. And something that you could do, and I think we should probably start doing, is going through ecosystems and building up genetic catalogs of everything, every single thing you can find, every plant, every animal, every fungus, all of it, and just building catalogs of it. So that way, when an ecosystem inevitably gets destroyed by human activity, whenever the humans are done ruining the area, when we want to put it back and make it nice again, then we would have a catalog where we could go, okay, just print out XYZ and bibbidi bobbidi, recreate all the different organisms and just put them back. It would be very, very difficult to do. It would be a huge undertaking, but at this point, I mean, the world is kind of only being left with difficult options. So it's kind of inevitable, so we might as well prepare for it. And this technology, this is essentially the only actual use of this technology that I don't think is incredibly dumb. Like, mammoth steaks sound really fun, but I'm much more interested in just repopulating walleye and the bees and all of the other, right? Like, this could be a job, right? Like, there could be, instead of the vanity companies like George Church's wanky nonsense, if you were actually just focused on rebuilding ecosystems and de-extincting the animals that had just gone bad or gone missing, I just think that there's so much more value in that personally, rather than like somebody's weird ego trip. And yeah, that's really just sort of the one way to look at it. And if we already have a seed vault, like, so this is the Svalbard seed vault, where tens and tens of thousands of different types of seeds are being saved, specifically to prevent exactly something like this from happening, which is when an ecosystem goes or is damaged, there is a repository of genetic material which can be used to regrow it. So in this case, you just plant the seeds. They're plants. You don't need to do any weird cloning to them. But I mean, seeds only last for so long. So you also have to be, regularly re-upping the seed vault and replacing the specimens that are getting really old, growing ones that have been sitting around for too long and producing new seeds. But if we have a seed vault, like, why don't we have a meat vault? Like to me, it just makes sense to have a facility somewhere where you can just maintain massive quantities of samples of all of the different fauna and flora from around the world, because thankfully humans are moving towards the direction of wanting to not completely obliterate the planet. And really even though we're headed in the direction of wanting to fix it, we're still actively doing damage. And, thanks to several corporations and, corrupt politicians and such, ecosystems are still going to get destroyed. And damage is still going to be done. And, developing countries are still going to develop. And so that's going to damage more ecosystems. And it's sort of inevitable. So it makes the most sense to me to prepare for the inevitability rather than just hoping that we're going to be okay or that we're not going to do it when we know that we will. So this way, threatened species are not that they could be ignored. They still need to be dealt with, but at least you have an emergency, break glass in case of extinction option, as opposed to just, good night, sweet prince, and off into the oblivion you go. So I think it's a very reasonable option, personally. Luca donated. Thank you, Luca. And I'm glad you enjoy the videos. And then the last option for this is seeding other planets. So, there's a lot of talk of terraforming Mars, which would be ungodly difficult and take 3,000 or 4,000 years. But shipping animals into space is not a great idea. We have enough trouble with, horses in trailers, not to mention horses in space. A panicking animal is not a thing that you want on a spaceship. But if you have artificial womb technology and just, a bunch of frozen cells, when you get where you're going, it would be much easier, or theoretically, if the technology was developed, it'd be much easier to just clone out a whole bunch of the animals that you want from a genetic stock that you bring along, as opposed to having to, bring the live animals either in stasis or awake through space. So it's just another option. But I think it's one of the few actually useful uses of the technology. But yeah, that's kind of it. Today we're keeping it a little bit short, insofar as I didn't want to get into the code this time because it feels a little bit cheap to just do one gene and put it into chicken or something. Mainly because it's not really de-extinction. A lot of the headlines were very clickbaity, and I really wanted to sort of talk about how clickbaity it is and the realities of the technology rather than kind of getting into the nuts and bolts of coding it. And also we got to talk about cloning, which I'm going to just go back up to the cloning section for a second. Scroll. Recently I was asked by someone, is there any technology that I feel is out of reach for me personally right now? Because the things that we have planned for the rest of the year, I mean, is just gonna I think it's gonna just blow all of your minds. Seth Love donated. Thank you, Seth. Glad you like the videos. But yeah, so the stuff that we have planned for the rest of the year includes growing organoids and vascularized organoids on a chip, and all this kind of stuff. So I really think that cloning is actually just about the only thing that's out of reach of us at this particular red hot second. But only barely. The biggest thing between us and cloning is legality, ethics, and money. It's just an extremely expensive process. So it could be but it's not impossible to say that 5 years down the line, when the channel is much bigger and we've got more of this stuff under our belt already, that it isn't something we could do. We could, if we got the right licenses and got the right approval and had all the right specifications. There's nothing actually stopping us from cloning something, be it a mouse or a pig or whatever. I don't plan on doing that, but it was sort of the only thing that I thought of. But so when you actually look into like how difficult cloning like is, it's not like that hard. It's mostly the reagents are really, really, really expensive because unsurprisingly, keeping eggs and these sorts of things and embryos growing outside of a womb requires a bunch of very weird specialized chemicals and reagents and proteins that are not cheap because why do you need a liter of like follicle juice? Like it's just a weird thing to want to buy. Yeah, it's pretty doable. It's just very expensive. Alrighty, so we're gonna end off with some question time. So I'll take some questions and then we'll wrap it up. I'm keeping it a little bit short today. I'm a little bit under the weather, so I don't want to stick around for too long. But also, I think this is kind of fun that we got to talk about cloning and chromosome printing and chromosome transplantation because it's a lot of stuff that you don't really get to see the nuts and bolts of it very often, and especially not in media or literature. All right, so question time. All right, let's see what we got. What is the minimum information required for this to work? Simply the DNA? Yeah, pretty much. If you have a complete genome sequence of something, theoretically you can de-extinct it. And even if you're missing chunks, if you've got the nearest relative, then you may be able to recreate some, like enough of the code that you end up with something functional, but then that gets pretty iffy pretty quickly. Okay, here we got a question from V Void. Would prion diseases be a worry with lab-grown human brain burgers? First of all, don't eat brain. Just don't. Just don't eat brain. It's not worth it. I don't care how delicious it is. Don't do it. It's not worth it. Like, prion disease is the worst way to go. So, yeah, don't. But, yeah, prion diseases wouldn't really be a concern with lab-grown meat that's done properly. This would have to be tested for, and it would have to be quantified. But if you're not growing brain tissue and eating brain tissue, it's not really an issue. Like, if you're just growing muscle, like, it's not a... it's not a particular concern. So yeah, it's it's not that bad. Okay, what else we got? Why is the limit 180 on DNA printing? The limiting factor is the chemistry. Currently, DNA is printed using the what's it called? The phosphoramidite process, where it's a series of chemical steps where you are protecting and deprotecting various parts of the molecule to progressively grow the chain. Over 180 letters or base pairs, you start getting more and more and more errors. Like the process, like the chemistry just gets more and more iffy. So they limit it to that because that's just sort of what the technique is capable of. The future of DNA printing is enzyme-based printing, but it just hasn't gotten to the same point yet. I saw a thing the other day where enzyme-based printing has gotten up to like 60 letters. Which is just not sufficient. The whole reason people were excited about enzyme-based printing is that theoretically you can get, thousands and thousands of letters printed all at the same time. But it just thus far hasn't materialized that way yet. But I mean, the technology is always developing, so eventually it will get to that point. And then the price of DNA printing will come way down. All right, what else we got? GSG, thanks for the content. You're very welcome. Glad you like it. What else we got? I'm gonna do a couple more, and then we're gonna wrap it up. When do you think we're gonna be able to make organisms with desired morphologies? Also, what do you think about artificial base pairs in xenobiology? Okay, so crafting an organism to be exactly the way that we want is like 100 years away, maybe more. Like, it's really hard. Designing an organism de novo to do something is very, 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 very difficult. Like, when it comes to DNA, right? If DNA is a language, we're at the point of maybe putting some words together into like a basic phrase. We are not at the point of writing books. Making an animal is writing a series. Like, it's not even writing one book. It's writing a dozen books. It's very, very difficult. Making small changes to organisms, that's easy. Making big changes, very hard, and usually ends in tears. So yeah, we're pretty far away from that still. Okay, what else we got? How does the cell seal with electricity? When you put the electrical impulse into the cell, it sort of makes all of the lipids and proteins and stuff align, and it changes the charge on the cell itself, which helps make everything kind of stick back together. The cell will naturally just close up the hole if you leave it alone. It has mechanisms for doing this. This just helps do it quickly and to make sure that you end up with the least number of exploded eggs as physically possible. And also, sometimes the electrical pulse isn't actually to seal the egg. Sometimes it's more to, rather than sealing the egg, it's actually to kickstart the growth process. So in Invincible, where they show the Mauler brothers electrocuting the cell and then it starts dividing, that's actually what happens. Sometimes you have to zap it and then it starts dividing as if it has just been fertilized. It's really weird. I don't know. I don't think anybody understands why that works, but it does, and so people do it. But yeah, it's definitely possible. Okay. Can you 3D print cell scaffolds? Yeah, sure. They do it all the time. There's lots and lots of literature on doing that. It's something I want to do within probably the next year or two, is 3D printing bone scaffolds and then growing like a set of antlers or printing a skull or something. Okay, I'm going to do one or two more, and then we're going to wrap it up for the day. Is it possible for plants like cycads, where only the female exists, to de-extinct the male? Probably not. I mean, unless you have if you have a sample of a male plant, then you can recreate the male chromosome, maybe. But yeah, it'd be very tricky if you don't. You'd have to find the nearest neighbor and like fudge it a little bit. But yeah, it would not be easy. Not be easy at all. What are cell scaffolds made of? That depends. It could be all kinds of stuff. Some people will actually grind up tissue, like they'll decellularize tissue to remove all the cells and leave the extracellular matrix behind, freeze dry it, grind it up into a powder, mix it with some like a gelling agent, and then like rehydrate it, and then just print that. Or they'll mix it with like a UV polymerizing agent, so that way it will they can resin print the scaffold. Sometimes even with the cells mixed in, which is wild to see. Do you know the mechanism used on enzyme-based printing? Yeah, so it's using an enzyme called TDT, I think. And then they're flowing specially modified substrate and enzymes over it like progressively to stick on one letter at a time. It's pretty difficult, but it is possible. But but beyond that, I don't know more of the specifics of the technology. I'd have to read up on it. But yeah, if you just look up enzyme-based DNA printing, you should find all kinds of results. Oh, okay. This is a really good one. To what point can you DIY your lab? My answer is as little as physically possible. Like, especially at the beginning of my career, like I was DIYing a lot of my equipment, and it just is not as good. Like, there's definitely certain pieces of equipment that you can DIY, but most of it you should just be buying. Like, it's one of those things, if you're either spending your time making your equipment or you're spending your time using your equipment, and you can't do both. So if you actually want to do biology, you should just buy your equipment. If you want to just build bio hardware, you should build a bio hardware. But that's kind of been my preference for that. Okay, one more, I think. Let's see if I can find a good one. Okay, this isn't going to be it, but I'll just answer this quickly. Is the stream saved as a video? Yes. These streams stay up, so if you want to go back and watch them later, they're available on the channel afterwards. All right, I'm just gonna scroll around a little bit and see if I find a really good question to wrap things up with. Actually, this is a fun one. Okay, how realistic or unrealistic is the use of ostrich eggs in Jurassic Park? Actually, it's pretty close. Like if you were gonna try and de-extinct a dinosaur. I mean, first of all, dinosaurs are not extinct. Like a lot of birds are technically classified as dinosaurs, like a chicken is. And there have been some studies, and I want to recreate this. So I actually have a piece of T-Rex bone. I didn't bring it here so that I could show it off on camera, but I do have a piece of T-Rex bone. And there has been a bunch of studies where, rather than trying to extract DNA out of T-Rex bone, they basically they stick it into a machine called a mass spectrometer. Or like they crack it open and they take a core sample, they stick it into the mass spec, and then they shoot it with a laser beam that boils off a little bit of the surface of the sample. And they actually found that there are protein remnants in the bones. And like they were able to recreate or partially sequence the collagen protein that was found in T-Rex bone. So there is actually soft tissue preserved after millions of years, which is counter to a lot of what people are, taught. There's definitely some debate over that particular bit of research. There's like a lot of talk about contamination and that kind of thing. But there's been a few studies that seem to point to that there is like detectable collagen and other proteins in these bones. So once you have the protein sequence, then you can just reverse engineer it, make it into DNA, and then start, one by one, putting those genes into a cell, which you could then use like an ostrich egg as a incubator of sorts. In the same way that you could use an elephant egg to grow a mammoth, you might be able to at least partially grow something a little more dinosaur-like in an ostrich egg. It would just be very difficult. Like I think it would probably take you 50 to 100 years to get this to work properly. But the biggest limitation is the lack of like the protein and DNA samples from dinosaurs. Like the samples are highly degraded. Even the little bits of protein that they were able to tease out are highly degraded. And when they were able to finally put it together into a full protein sequence, it was essentially identical to chicken collagen. So like T-Rex collagen and chicken collagen, as far as anybody can tell, is essentially the same protein and it's been completely unchanged over millions of years. So it's very cool and it's very weird, but at the same time, it's not enough to build an entire T-Rex. You're still missing most of its genome, the overwhelming majority. So you could start to tease out details, but you're never going to get a Jurassic Park. You could get like an Ice Age Park. Like Ice Age Park could totally be a thing. The nice thing is a lot of those animals died in ice. So you can just go dig them up out of the permafrost if you get lucky, and then you have all the DNA that you could ever want. But dinosaurs are just so damn old that it's just essentially impossible. However, I mean the main concept in Jurassic Park of finding mosquitoes and getting the blood out of the mosquitoes is less ridiculous than you'd think. I don't think the DNA would be preserved properly, but if there was ever a chance to preserve proteins, that might actually be it. The sequences are going to be so heavily degraded that actually like resurrecting an entire dinosaur is just not going to happen. All right, I think I'm going to do one more. Like I keep saying, I'm going to do just one more. Are there any other good questions? Otherwise, we're gonna call it for a day. Somebody said they used all kinds of different eggs in Jurassic Park depending on the size of the thing. Yeah, I mean that makes sense. If you're making a small dinosaur, use a chicken. If you're making a big dinosaur, use something bigger. It's okay. So what else we got? Do we have ice preserved saber tooth? I have no idea. Not a clue. But even if it's not ice preserved, even if you've just got the bones, it is possible to extricate DNA samples. Like this is how we've been able to build the DNA samples of Neanderthals and Denisovians and other ancient hominids and other ancient species. Like as long as it's less than 50 to 100,000 years, you can tease out some of the DNA. The trouble with Neanderthals is that our DNA and their DNA is so close that it's very difficult to tell which is which, because there was a lot of like interbreeding. Like we were technically different species, but not by like the way we define species, because you shouldn't have been able to breed if we were truly different. And yet, a lot of our DNA is their DNA, so it wasn't really that they went extinct so much as the species kind of fused violently. So it's a bit of a mess. But if you have saber-toothed cat fossils, or not fossils, but like specimens, the dental pulp is actually one of the best places to get DNA from. And they actually, so in Egyptian mummies, one of the best places to get sequenceable DNA out of an Egyptian mummy is the dental pulp. So you pull a tooth, and then right in the core of it, there's some soft tissue, or there was some soft tissue, that is basically protected from all of the stuff that happens during either mummification and also the thousands of years that the corpse is left to sit in a tomb somewhere. But the dental pulp leaves it really quite well preserved. So the way that we've been able to figure out, there were a couple of mummies that were found that were suspected to be like Nefertiti or one other famous mummies. This is sort of how they're starting to tease this out, which is these dental pulp extractions. And if you have a saber-toothed cat and it's in good enough condition, theoretically you might be able to get enough of the DNA out of it that you could de-extinct it if you were so inclined. But it would be, again, I mean, all the de-extinction stuff is just hard. Like, none of it's impossible. It's just really, really, really hard and really, really, really expensive. And a little bit pointless, other than it's awesome. Like at the end of the day, it's like, why would you de-extinct a mammoth? I don't know. It's cool. You can ride a mammoth around. But I mean, it's why do we do anything? Why do we build fast cars? Why do we build rockets to go to other planets? Mostly because it's cool. Like, like the Discovery Channel or NASA or whatever will like bill it as, a search for discovery and the greater humanity. If you ask any of the engineers, why did you build the giant rocket? They're not going to go, because I want to do they're like, I wanted to make the giant thing go kaboom. Like, most scientists don't we like to performatively say that the reason that we do science is like the betterment of humanity and stuff. 99% of the time it's because it sounded fun or it sounded cool. It's like, why did you de-extinct a mammoth? I don't know, it sounded fun. Like, it's at the end of the day, like, all of these sorts of things you do because it's cool. Like, some people, like, unless you're actually working on something that will dramatically help the population, like, making wheat the size of corn, like, you're just not going to, you're like, most of the things you're doing in science are because it sounded fucking awesome. And, you're going to be spending the next 5 to 12 to 15 years of your life on it. So you might as well enjoy it. Like for me, like, why do I grow neurons? Like, why do I want to grow neurons and have it play Doom? Because it sounds cool. like, yeah, it's actually going to, like, if we manage to get neurons to play Doom, it will legitimately be a breakthrough. It will be the most complicated thing that anybody's ever gotten neurons to do. But that's not why we're doing it. We're doing it because it sounds fun. And it's like, it's why not? So that's kind of where you go with that. Okay, let's see if we can do one more, and then I really gotta wrap it up. Okay, what sequencing services do you recommend or use? The only service that I use at the moment is Plasmidosaurus, but that's only for sequencing plasmids. I don't do a lot of full genome stuff, so it's just not something I've had to worry about. Okay. I think that might be it. I think we're out of questions for the day. So yeah, I think we'll end it there. We got a lot of fun stuff coming up on the channel. So if you haven't already, be sure to like the video, subscribe, all that kind of stuff. The next couple of months I think are going to be a lot of fun. We're trying to get a video out for the end of this month, but it might be out sort of beginning to middle of May. We'll see. But we'll be growing some like mammalian cells and genetically modifying them. We have the neuron video coming out very soon, where we're going to be like growing our first batch of neurons on these beautiful brand new electrode arrays. This is another little thing that I brought in. It's really hard to see, Yeah, doesn't show up well on camera. But this is a laser cut test. So part of making these new neuron arrays involves using a fiber laser to cut very, very tiny holes. So all of that's coming in a new video very soon. I'm very excited for it. We've got a lot of neuron stuff this year. We've got a lot of mammalian stuff this year. We've got lasers. We've got high temperature stuff. Just a lot of stuff. So yeah, if you're interested in all of that, highly recommend subscribing. But other than that, I'm going to leave it there. I hope you guys have enjoyed. This has been a lot of fun. There'll be another stream either end of this month or around the same time next month. We're sort of figuring that out. We still want to do the million subscriber celebration stream because we've blown right past 1 million subscribers way faster than I thought we were going to, and before we got a chance to do the celebration stream. So we'll come back to that very soon. But yeah, other than that, I hope you guys have enjoyed. It's been a lot of fun, and we'll see you on the next one. Have a great day. Bye.